Okay, good morning all students. Today I'm going to discuss about the host microbe interactions and what are basically the host microbe interactions. Today I'm just going to cover about some basics as well as the definitions of pathogenicity as well as infection and the virulence. Okay, these three terms will be discussed today along with the basics of the host microbe interactions. Okay, the host microbe interaction which we are going to discuss is basically of a parasitism. Okay, is basically of a parasitism. In the case of a normal human microbial flora, okay, where two interactions were commensalism and symbiotism or mutualism more specifically was basically discussed. Okay, which are the microbes uh, which live in a human being, but in this case we are going to study the microbes and the mechanism of the microbes which cause disease in the human beings. So starting with the host microbe interactions. Okay, uh, as a whole we are going to discuss these points not in today's lecture, but as we move forward with the host microbe interactions, starting with the process of infection, pathogenicity, virulence and infection, microbial adherence factors, penetration of epithelial cell layers, events in infection following the penetration into the host, and microbial virulence factors. Okay, today in our today's lecture we are going to discuss about the pathogenicity, virulence and infection, and in tomorrow's lecture we are going to discuss about the process of infection. Okay, so moving forward with the basics of the of this unit. Okay, a disease, what do you mean by a disease? Okay, disease is basically a condition in which a normal structure or the functions of the body are damaged or impaired. Okay, please write down this definition. Okay, a disease is any condition in which any normal structure or the functions of the body are damaged or impaired. Okay, and the reason, whatever may be the reason, sometimes it may be due to infection, sometimes it may be the case where you are hurt, and after you are hurt you may get a infection due to which a disease is caused. But practically, okay, physical injuries and disabilities are not classified as disease. Okay, please note this point also. But there are several causes of the disease, including the infection by a pathogen, even the genetics. Okay, for example, uh, various diseases such as diabetes. Okay, as well as some of the cancers, which is also a diseased condition, and non-infectious environmental factors. Okay, and as well as inappropriate immune responses, right? That is in the case of autoimmune disorders. Now in the case of infectious environmental causes, it may be pathogen also, it may be unfavorable conditions of the environment also, or whatever the conditions may be. And the pathogens basically includes variety of, it may be bacteria, it may be viral, okay, or it may be even any of the worms also, even, okay, even worms also. Okay, they are also responsible for causing a various disease. Okay, so now the next term is infestation. The next term is called infestation. Many of the people in the scientific world, basically many of the students, even many of the faculties I have seen, they are, when they discuss infection, okay, when they discuss infection, okay, not infestation but infection, they said that large parasites, okay, arthropods which enter our body are basically called as infection, but it is actually not true scientifically. The scientific word used for the presence of large parasites like worms, insects, or even the larvae of the insects, okay, or any other organism, okay, which are basically macro ones which live inside the body. The word used in that case is not infection, but it is called infestation. It is called infestation. Do not confuse infestation with the infection, okay, because their mechanism is different from the microbes, okay, which cause infection. Their mechanism is different, okay. Now moving with the signs of a disease. Signs of a disease. Signs of a disease are objective and measurable and can be directly observed by a clinician. Okay, let's take an example. Let's take an example of Gautam. Okay, Gautam Mehta, and let's say he has some fever, whatever. His health is not doing well. Okay, so he may have some problem, such as which can be measured, such as his body temperature is high. It can be measured. Okay, his breathing rate is high, it can be measured. Okay, so these are basically the signs of a disease. Okay, let's take an example. Now Gautam Mehta may be having a hepatitis. Okay, his body will turn slightly yellowish. So yellow color of the skin indicates a sign of the hepatitis. Okay, indicates the sign of a hepatitis. Okay, so the science basically help the clinicians or the scientists or the doctors to identify a disease also, and also check the various parameters which are indicative of a disease. Even it gives us an idea which organ may be infected in our body. Okay, so vital signs which are generally used to measure the body's basic functions include body temperature, okay, which is normally 37 degrees Celsius, and in Fahrenheit it is 98.6 Fahrenheit. Okay, heart rate which beats normally between 60 to 100 beats per minute, and normal breathing rate is around 12 to 18 beats per minute, and the blood pressure which normally around 90-60 and 120 to 80 millimeter, so mmHg mercury. And changes in any of the body's vital sign may be indicative of a disease. Okay, there are also chances due to any injury or any other chances, these vital signs may be also seen. Okay, may be also seen in the body. For example, when we are discussing a disease, for example, having a fever, okay, a body temperature significantly higher than 37 degrees Celsius is a sign of a disease. Okay, why? Because it can be measured. Because it can be measured. Okay, now moving with the next term, symptoms of a disease. Okay, symptoms of a disease. Okay, as the signs were the objective ones, whereas the symptoms are basically the subjective ones. Okay, signs were the objective ones and the symptoms are the subjective ones. Symptoms are basically felt and experienced by the patient. They cannot be clinically confirmed or objectively be measured. Okay, these are the feelings or the experience by the patient, okay, and which includes nausea, okay, loss of appetite and pain, okay, and such symptoms are important to be considered when diagnosing a disease, but they are subject to memory bias also and are also difficult to measure precisely. Okay, and some clinicals do try to attempt to quantify symptoms by asking the patients to assign a numerical value to their symptoms. Okay, uh, basically clinicians may try, okay, to find out what actually is the problem. Okay, so what is nausea? What is nausea? Okay, nausea basically is a stomach discomfort. Okay, it's a stomach, there is a problem in a stomach, okay, which will give you a sensation, okay, like you want to vomit, okay, like you want to vomit. And nausea can be precursor to vomiting, the contents of the stomach also. And the conditions may arise, okay, may arise with a variety of causes, not, it may be not even a, it may be also a single cause, or it may be a variety of causes also. Okay, and in every cases it's not because that nausea may be only caused during a disease, it may also arise uh, from another conditions with, where you do not have even disease, such as if you are in car, okay, sometimes, okay, which include motions, even if you are in a aeroplane, where in the case of a landing, in a ferries, okay, in a ferries, as well as when you drink too much of alcohol, okay, too much of alcohol drinking, as well as you may, it may also arise when you basically eat a lot. Okay, there are various other causes, okay, which are present in the case of a nausea. And yes, nausea signs can be identified, signs of nausea can be identified. Okay, nausea is a symptom, but its signs can be identified, like restlessness. Okay, restlessness, it's like you do not feel well. Okay, you feel, yes, I need to do, I'm not feeling well, I'm feeling tired, or something like that. Okay, fatigue, okay, increased in salivation, sorry, increase in the salivation also is one of the symptoms, sorry, is one of the signs of the nausea. Okay, urge to vomit, okay, you will have something like, yes, I'm going to vomit, yes, I am going to vomit, you have something like that of feeling. Okay, okay, loss of appetite, your food consumption, okay, your food consumption will be lowered. Okay, and headache also, dizziness, even sometimes even sweating. Okay, and sometimes even the abdominal pain, may have pain in your abdomen or stomach particularly. Okay, so this symptoms basically can be, if some have signs, can be identified, but are basically characterized on the on the on the questions which are asked by the clinicians to the patients. Sometimes there are chances, if not answered properly, the diagnosing may become difficult in the case of a symptoms. Okay, and these are some of the examples of symptoms when you may have a inflation. Okay, when you may have a inflation, which includes fever, pain, soreness, swelling, edema, granulomas, abscess, lymphadentis, lesion. Okay, these are the conditions where you may have a inflation. Okay, now what do you mean by edema? Okay, basically edema is a medical term, used for swelling. Okay, edema is a medical term used for swelling. Okay, where body parts basically body parts start swelling due to the injury. Okay, or due to the inflammation as well. Okay, or due to the inflammation also. Okay, and sometimes other conditions, okay, which are not disease conditions, edema may also occur. Okay. Now moving forward with the granuloma. What do you mean by granuloma? Okay, granuloma is simply uh, a type of inflammation. Okay, it's a type of inflammation of a very small area, and are found uh, in a conditions, okay, in a conditions of many diseases as well. Okay, but it can be identified by the collection of immune cells known as macrophages at the given point of granulation. Okay, and granulomas form when the immune system attempts to all of substances it perceives as foreign but is unable to eliminate. Okay, but is unable to eliminate. Okay, immune system will try to remove the foreign particles which are harmful, but it is not able to remove this. Okay, uh, remove the foreign particles, due to which a granuloma may be formed. Okay, lesions you already know. Now these are some of the common signs and symptoms of a infectious diseases. Okay, signs: fever, septicemia. Okay, fever, septicemia. Okay, next comes microbes in fluids. Okay, next comes microbes in fluids. Okay, next comes chest sounds, next skin eruptions, next leukocytosis, next leukopenia, next swollen lymph nodes. Okay, next abscess, next tachycardia, also known as basically increased heart rate. Okay, as well as the antibodies in the serum. Okay, now what do you mean by septicemia, also known as sepsis? Okay, also known as sepsis. And septicemia is basically a serious bloodstream infection. Is basically a serious bloodstream infection, okay, which may be due to the blood poisoning. Okay, blood poisoning. Okay, and it occurs when a bacterial infection, okay, occurs in the body. Okay, and such as lungs, skin, and the bacteria enters the blood. Okay, and this is because of the bacteria and their toxins which are produced by the bacteria by the bacteria. Okay, now sepsis, it may also occur due to the some of the chemical substances, okay, which are also harmful. Okay, now the next term, the symptoms, which includes chills, pain, ache. Okay, means you may have some time pain, malaise, soreness, irritation, fatigue, chest tightness, itching, headache, nausea, abdominal cramps, anorexia, like you have a lack of appetite, and the sore throat. Okay, these are the basically symptoms. Okay, now what are the info? Why do we need to study these terms? Okay, it is important because we may get an idea what kind of infection or what is the organ of the infection. Okay, and the leukocytosis, which we can measure, is basically a condition, okay, is basically a condition where there is increase in the number of white blood cells, increase in the number of white blood cells. Okay. Now leukopenia. Next term is leukopenia. Okay. Leukopenia, basically it's a condition, okay, where you have a lower count of white blood cells, where you have a lower count of the white blood cells. Okay. Now moving forward. Now basics. Now we have already discussed symptoms, signs, and what is disease. Now moving forward with the syndrome. Okay, what is syndrome? A specific group of signs and symptoms characterized of a particular disease is called a syndrome. Okay, simply the word also used for the syndrome, we may also use the word group of diseases. Okay, we may also use the word group of diseases. Okay, it's also called as syndrome. Okay, and basically syndromes are named using a nomenclature based on the signs and symptoms or the location of the disease. Okay, it may depend on sign or the symptom or the location, also on various other factors also. Okay, now what are the nomenclature of the symptoms? How do we divide or different types of symptoms? Oh, I have given some of the some of the regular examples, common examples of the symptoms. Okay, where starting with the first one, the affix cyto. These are the affixes used, and the meaning is given here. An example is also given here. Okay, the cyto, the affix cyto, it indicates cell. Okay, and the cytopenia means the reduction, it means the reduction of the blood cells, it means the reduction of the blood cells. Next affix is the hepat, it means of the liver. Okay, it means of the liver, and the example is basically hepatitis. Hepatitis, it indicates inflammation of the liver. Okay, and the itis is basically here. Okay, itis, it indicates inflammation, it indicates inflammation. Okay, and now the second one, the pathy. Okay, now the second one is basically apathy, it means disease, it means disease. For example, neuropathy, a neuropathy, it means a disease affecting the nerves, a disease affecting the nerves. Okay, the next term is emia, the affects emia, it indicates of the blood, it indicates of the blood. Okay, bacteremia, bacteremia, it means the presence of bacteria in the blood, the presence of bacteria in the blood. Okay, next comes the term lysis, next comes the term lysis. Okay, it means destruction, hemolysis means destruction of red blood cells, the destruction of the red blood cells. Next term is basically oma. Okay, it means tumor, it means tumor. Okay, example is lymphoma, it example is lymphoma, the cancer of the lymphatic system, the cancer of the lymphatic system. Next one is oocyst. Okay, it is a diseased or an abnormal condition, diseased or an abnormal condition. Okay, example leukocytosis, example leukocytosis where there are abnormally high number of white blood cells. Okay, where there are abnormally high number of white blood cells. Now the last one is derma. Okay, last one is derma. It means of the skin. It means of the skin. Keratoderma means a thickening of the skin. It means a thickening of the skin. Okay. Now there are also various other terms. I will just given you an example of a few of them. Okay. Now the other basic terms. Okay, which includes a adherence. Okay, adherence, or shortly or easy word used for is basically adhesion or the attachment. Okay, it's a process by which bacteria stick to the surfaces of the host cells, and after the bacteria have entered the body, the adherence is the major initialization step in the infection process. The first step of basically infection is the attachment or the adherence. Okay, the term adherence, adhesion and attachment are often used. Okay, are often used in the case of adherence. So basically adherence is a process by which a bacteria or any microbes basically attaches to the surface of the host cell. Okay, sometimes the word adherence, sometimes the word adhesion, or even the word attachment is used. Okay, now the second terms comes is basically carriers. I'm going to discuss carriers in the next lecture in more detail. Right now I'm just going to finish it with the definition of the carrier. A person or animal with asymptomatic infection then can be transmitted to another susceptible person or a animal. Okay, where asymptomatic is with, okay, sorry, a carrier. For example, if a Gautam is a carrier, okay, is a carrier of, let's say, any of the disease. Let's take an example of a typhoid. Gautam is a carrier. Okay, he does not have any symptoms. He may have an infection, but they are not symptomatic. Okay, but they are not symptomatic. But he can transmit this disease to another person also. Okay, for example, he comes in contact with the sagar. Okay, there are chances that he may transmit the typhoid with to the sagar. So in that case, the Gautam is basically a carrier. Okay, the word next comes asymptomatic. The word next comes is basically a asymptomatic. Is basically a dimmed infection. Okay, what do you mean by dimmed an infection? It means there are very less number of symptoms or no symptoms appear. Okay, in that case, but he will be having a pathogen in his body. Okay, now the next term comes is invasion, the process whereby bacteria, animal parasites, fungi and virus enter the host cells or tissues and spread in the body. Okay, simply it's a process for bacteria, various parasites or viruses enter the host cells or tissues and spread in the body. Okay, now the word pathogen. Okay, I'm moving towards the pathogenicity. Now I'm moving towards the pathogenicity. First we need to understand the term pathogen. Okay, first we need to understand the term pathogen. A microbe whose relationship with its host is parasitic and results in infection and disease and a disease. Simply it's a relationship between two individuals which have a parasitic type of relation, okay, which have a parasitic type of relation, okay, and can cause disease in the host. And the type and the severity of the infection basically depends on the pathogenicity, of the organism and the condition of the host also, okay, and the conditions of the host. So there are variety of conditions, such as the whole condition is basically age of the host, even the immune system condition of the host, okay, also depends on the diet, also depends on the environment. So there are variety of conditions, okay. He may also be on the medication, he may also given even vaccine. In that case also it depends, okay. Simply, if you want to write in a small sentence, a microorganism which is capable of causing a disease is called a pathogen. A microorganism which is capable of causing a disease is called a pathogen. Now, some of the specific which I am going to discuss today, okay, is basically pathogenicity, virulence, and infection. Okay. So the pathogenicity. What do you mean by pathogenicity? An organism's potential to cause infection or a disease, okay. It's simply an organism's potential to cause infection and cause infection or a disease. There are basically two types of pathogens. One of them is true pathogens, okay, and other ones are basically opportunistic pathogens, okay. So what is the difference and what are the characters, why do we and how can we basically differentiate with them, okay. There are other various other points where the pathogenicity can be further related, and basically depends on the virulence also. And the virulence is basically the degree to which an organism is pathogenic, okay. It's a degree of pathogenicity to which, okay, a, a organism is virulent, okay. Basically, for example, there are some microbes, okay, for example rickettsia. They are obligate parasites, okay, which are going to cause a disease. Another example is of a Mycobacterium tuberculosis. It is going to cause a disease, okay. Now comes the opportunistic pathogens, which includes Escherichia coli. It is not going to cause a disease in a regular conditions. It is an opportunistic pathogen, okay. Another example is often Candida, okay. It will only cause a disease when there is a change in the normal microbiota, okay. So what are the two major differences between a primary pathogens and the opportunistic ones? What are the differences between them, okay? The first and the basic foremost being the primary pathogens is a regular cause of what the disease, okay. They are going to cause a disease, whereas the opportunistic ones takes the opportunity offered by the host defenses, okay, offered by the reduced host defenses to cause a disease, okay. So it depends. Opportunistic pathogens will only attack the body when the host defense system is lowered, okay, when the host defense systems are lowered, okay. So the next term is a long term survival of a microbe is the another main difference, okay. Long term survival in the case of a primary pathogen is absolutely dependent on its ability to replicate and be transmitted in a particular host, Okay, so in the case of permanent pathogen, how much time it will be in the body purely depends on its ability of a pathogen. For example, Mycobacterium tuberculosis can stay in a human body for a very long time, for example 6 months, 1 year, depending on the severity of a disease. Okay, malaria parasites can be found for over 2 months also in our body. Okay, depends again on the medications and uh, other information which are sorry, other problems in the basically host. Okay, other conditions of the host as well. Whereas in the case of an opportunistic pathogen, okay, it may differ to the case of number of pathogens that infect humans. Okay, for example, the E. coli, it basically resides in a human for a very long time. Okay, but it may not cause a disease, it may not cause a disease for a longer time, but when there are higher chances when the host immune system is basically defense systems are lowered. Okay, in that case only it is going to cause a disease. Okay, now moving forward with the next term is basically virulence. Okay, is basically a virulence, a degree of pathogenicity determined by its ability to establish itself in the host, to establish itself in a host and cause damage, and cause a damage. Okay, now the virulence factor. Okay, virulence factor, any characteristics or structure of the microbe that contributes to its virulence, that contributes to its virulence. Okay, is basically a virulence factor, is basically a virulence factor. And the different healthy individuals share varying responses to the same organism. Okay, why? Simple evolution, simple evolution. Host also evolves, host also evolves. Okay, and there are also various other conditions like geographical conditions, even the racial conditions, which are going to have an effect on the virulence power of the organisms also. Okay, simply, okay, for example, on one end of the spectrum are the organisms that are basically virulent. On other or another organisms, okay, that are highly virulent. Okay, and highly virulent pathogens will almost always lead to a disease when introduced to a body, and some may even cause a multiple organ or a body system failure, even in the case of a healthy individuals. Now the less virulent pathogens may cause initial infections but not always a severe illness. Okay, and pathogens with low virulence would more likely to result in the mild symptoms as well as signs of a disease, such as low grade fever, headache, okay, and the muscle aches. And some individuals, in the case of some symbols individuals, it may be also asymptomatic. So in the case of a pathogens with a low virulence power, there may be some signs and symptoms, okay, which are mild ones, but in some cases they neither of signs nor symptoms will appear, neither signs nor symptoms will appear. Okay, for example, now I am giving you an example of a highly virulent organism, okay, which is a basically let's take an example. Okay, Bacillus anthracis. Okay, Bacillus anthracis, you all know, it's an organism responsible for the anthrax. Okay, it's a pathogen which causes anthrax in human beings. Okay, so the bacillus anthracis can produce different forms of a disease depending on the route and the transmission. It may enter through the skin or inhalation or the even the ingestion also. Okay, and one of the most serious form of the anthrax is basically inhalation. Okay, and the bacillus anthracis spores are basically inhaled and they germinate and active infection develops and the bacteria release potent toxins which cause edema. Okay, I already told you what do you mean by an edema. Okay, an active infection basically develops and the bacterial release potent toxins which cause edema. It's simply if you want to say a fluid will be built up in the tissues and which may lead to hypoxia like conditions. Okay, which may lead to a hypoxia like conditions. Okay, um, uh, do you know what do you mean by hypoxia? Okay, let me tell you in a short. It is a condition where oxygen is prevented from reaching the tissues. Okay, it is a condition when oxygen is prevented from reaching the tissues. Okay, and sometimes it may also lead to the necrosis. Okay, it may also lead to necrosis and which finally leads to the cell death also. Okay, so the simply if you want to list out signs and symptoms of a anthrax, it includes basically high fever, difficulty in the breathing, okay, even the vomiting, coughing of a blood, and sometimes chest pains also. Okay, so while inhalation of a anthrax toxins and the bacteria enter the bloodstream, which can lead to multiple organ failures, okay, and even the death of a patient. So now if the gene which is basically involved in the pathogenesis is inactivated, the same bacteria bacillus anthrax can become less virulent or non-pathogenic. Okay, now if the same organism now tries to infect a human being, it will not be able to infect. Okay, it will not be able to infect, but there needs to be a system where we want to find out in a quantitative terms how virulent a pathogen is. Okay, you must may have heard a term, this bacteria are more virulent, these are less virulent. Okay, on which basis they are termed more or less virulent, on which basis? Okay, so it depends on the type of condition and other factors also. What are the conditions of the virulence? Okay, now it can be also uh, the conditions which also lead to various other conditions that it may or may not cause a disease. It can be measured. Okay, it can be measured by using the lethal dose or an infectious dose. Okay, what is lethal dose and what is infectious dose? Now coming to that point, the virulence of a pathogen is usually measured by determining the LD50. What is LD50? It is called as lethal dose 50. It is called as lethal dose 50 for a particular type of a laboratory animal. Okay, and it is defined as the number of organisms which when administered to a number of laboratory animals will kill 50% of them. Okay, will kill 50% of them. Okay, so it is basically amount of organisms or basically pathogens needed, okay, or sorry, when they are administered to a number of laboratory animals, it will kill 50% of the laboratory animals. It's called as lethal dose. For example, LD50 dose of a 10 cells of a strain compared to the 1 lakh cells of a strain Y would indicate that strain X is 10,000 times more virulent than Y. Okay, is more virulent than Y. Okay, I'll give you an example. It will be easy for you if I give you an example in this case. Okay, so now simply moving forward now, right now, the LD50 dose can be determined, okay, can be determined more precisely, than other endpoints, okay, such as dots that kills 100% of the animals, which is also known as LD100 dose, and sometimes it is also termed as lethal dose because the rate of change in the mortality versus the change in the dose is the greatest around the point of 50% mortality. Now moving forward with an example of the same. Okay, now how can we calculate it? There is another also term which is called as infectious dose 50. Okay, so ID50 is the number of pathogen cells or variants required to cause a active infection in 50% of the inoculated animals, and the LD50 is the number of pathogenic cells, variants and amount of toxin required to kill 50% of the infected animals. So infectious dose is the ability to cause infection, whereas the LD50 is the ability to kill the 50% of the infected animals. This is the basic difference between these two terms. Okay, LD50 indicates the ability of an infection. LD50 indicates how many of which 50% of the infected animals are killed. Here all the animals are basically experimental animals only. Okay, so to calculate these values, each group of animals is inoculated with one of the range of the known numbers of pathogens and cells or even variants. Okay, a known concentration or known numbers of pathogens and variants are administered into the animals, and the percentage of animals that have been infected for ID50 or killed for LD50 can be plotted against the concentration of a pathogen inoculated. Okay, so basically the determination, a determination of ID50 of a pathogenic organism. Now this one is an graph for the ID, how we can identify a ID50 for a particular organism. Okay, various doses of a specific pathogen are introduced into an experimental host. Okay, strain A is more virulent. Strain A is more virulent. Okay, now how? So starting with the at a 0 point, okay, where a dose will be administered into a experimental host animals, what will happen? Infection will start. Okay, but the number of dose per unit of time, okay, and the number of organisms will be into 10 to power 3, starting with 1000 and the 2000. Okay, for example, at a point 3, okay, at a point 3 where you can see that the strain A, okay, when reaches around this point, the 3 point time, you can see the percentage of infection is 50%. Okay, so we can see that the ID50 will be 3000. Okay, will be 3000. It indicates 3000 units of an organism. Okay, Now in the same scenario of a strain B, where it needs about 5, okay, 5 means 5,000, okay, it when they are infected with 5,000, it indicates 50% will be infected. Therefore ID50 will be 5,000 for strain B. So simply we can conclude that the strain A is more virulent as compared to strain B, because strain A needs very less amount of organisms to infect a particular individual, okay, to infect a particular individual, okay. So now moving forward, okay, so now moving forward with the next example of the ID50s, of the selected, some of the selected foodborne infections, okay. So okay, I'm going to give you one more example, uh, uh, which is basically of a other bacteria, okay. For example, that the infectious dose for an individual may vary, okay. Infectious dose for an individual may vary from individual to individual depending on factors such as route of entry, age, even the health, as well as the immune system. And there are also various other factors also, such as environment, pathogen specific factors, such as susceptibility to the various conditions of the body also. For example, I am giving you one example. A single cell of Salmonella enteritica, a serotype, can serotype basically typhimurium, which can result in an active infection. Okay, just a single cell. Okay, just a single cell. Okay, and the resultant disease will be Salmonella gastroenteritis. Okay, which can cause nausea, vomiting, diarrhea, and but has a mortality rate less than 1% in the healthy adults. So in contrast, Salmonella enteritica serotype typhi, which has a higher infectious dose and typically requires as many as 1000 cells to produce infection. However, in this case, the serotypes causes typhoid fever, but the problem now comes in this case, where the mortality rate in the case of typhimurium was hardly 1%, but the same with a higher ID50 dose, which requires more organisms to cause disease. But in the case of serotype typhi, the mortality rate is higher as about 10%, as high as about 10%. Okay, so this is the difference. It does not, ID50 just gives us an idea how many amount of organisms will be needed to infect an individual, but it does not gives an idea, okay, how it will behave in different types of individuals. But there are chances generally, for example, lower ID50 indicates a higher virulence power, but it depends purely on the environmental and other conditions of the host, as well as various other conditions also. Okay, for example, if you are in having a already taking up a medications which are preventive of a typhoid fever, you may not get infected. Okay, so these are the conditions which are going to uh, decide. Okay, so I'm just reading out these examples right now. Hepatitis A, ID50 is around 10 to 100. Escherichia coli, enterohemorrhagic, hemorrhagic, 10 to 100 again. E. coli enteroinvasive EIEC 200 to 5000. E. coli enteropathogenic, okay, more than 10 millions. Next one is E. coli enterotoxigenic, okay, again more than 1 million. Okay, Salmonella typhi around 1000 and Typhimurium around 1. Shigella dysentery 10 to 200. Vibrio cholerae around 10 lakh, sorry, around 1 lakh. Okay, Vibrio parahemolyticus, okay, around 10 or around 10 million. Okay, now I'm going to end my lecture right now. Okay, please join our session. Please join our session of the WebEx. Okay, and please